Thank you so much for doing this.
Anytime.
Okay, I think we'll get started. Welcome to the Centers of Excellence in Hematology Microscopy Mondays. We just want to give you guys a couple of heads up. This is our last of the series in Microscopy Mondays, but all of the Microscopy Mondays are recorded and made available on our website, which is CCEH.io. And if you just click on the tile that says Microscopy Monday, you'll be linked to go find all of the videos that have been recorded and will be available on that website. But today we're excited to have Daniel Lucas, who comes to us from the Cincinnati Children's Hospital Center of Excellence in Hematology. And you can see the title of his talk. We really appreciate you, Daniel, for speaking with us today. We'll turn the time over to you in just one more second. For those of you who are joining us, Daniel has agreed to questions throughout the talk, so you are welcome to unmute yourself and ask a question when it comes up, or you can populate it in the chat, and I'm happy to ask the question for you if that's preferred. Thanks everybody for joining, and we'll turn the time over to Daniel. Thank you.
Thank you, and thank you for everybody attending. What I'm going to focus on this seminar is describing how we are using conventional confocal microscopy to interrogate the bone marrow and to map production of blood cells in the marrow. So just a quick reminder, every second the bone marrow produces millions of blood cells, mostly myeloid cells, with a few lymphoid cells, because this needs to be produced at different ratios. This happens through hierarchical sequential differentiation, where you have stem cells at the apex of the progeny, and they are progressively going to differentiate, make lineage decisions, commit to a specific lineage, and eventually generate mature blood cells. This system is extremely plastic, and in response, for example, to hemorrhage, you're going to have increased erythropoiesis, inhibition of lymphopoiesis. In response to infection, you're going to increase production of monocytes, dendritic cells, and neutrophils at the expense of other lineages. And of course, the most dramatic example of plasticity is stem cell transplantation, where a single stem cell can home to the bone marrow, engraft, and regenerate a healthy system. I don't need you to remember any of these steps of differentiation. Just remember that this is hierarchical sequential differentiation by making fate decisions. All of this is happening in the bone marrow and supported by the bone marrow microenvironment. There are many components of the microenvironment, but the key anatomical regions of the marrow are going to be the bone surface that fully encloses the marrow, with arteries penetrating through this bone, giving rise to a very dense network of sinusoids that essentially covers the marrow. And this is both whole in place and tightly interacting with a network of reticular stromal cells. Hematopoiesis is going to happen in the interstitial spaces left by these major structures.
Hey, Miss Anna, how are you?
Sorry, is there a question? Okay. The field has tried to map hematopoiesis for decades, and this has been very challenging, and I'm going to enter in those challenges in detail in a few minutes. But from the seminal finding by Morrison's of the markers that allowed imaging of stem cells to other people have developed markers to study heterogeneous populations of erythroid and myeloid progenitors, as well as either single lymphocyte progenitors or pairs of cells highlighted here in yellow. And when we summarize these findings and look at a region of the bone marrow, we know that myeloid stem cells localized near megakaryocytes, whereas lymphoid bias stem cells localized near these arteries. Common lymphoid progenitors also have a preference for arteries, whereas erythroid progenitors and myeloid progenitors were thought to be more or less distributed through the marrow, and it was not clear if they were associating with specific structures other than the perivascular stromal cells. My lab tries to understand what is the physiology of hematopoiesis in the marrow and how the bone marrow functions as a tissue. And more specifically, how does the microenvironment regulate normal and stress-driven cell production? And to me, something that always frustrated me when I was training was that hematopoiesis has always been studied ex vivo. You purified your cells from a mouse or a patient, do flow cytometry analysis, and then do either in vitro assays to assess function or transplantations to quantify the output of the cells, or omics. But in all these experiments, there is a loss of spatial information. And this is because traditionally it has been very, very difficult to image the bone marrow. You have the softest tissue. The marrow is essentially a soup of cells that are loosely attached to each other and enclosed by the hardest tissue, the bone. And the field has to develop many different methods to try to interrogate this. The second problem is that many of the antibodies used to identify the progenitors using flow cytometry don't work for imaging, either because the epitopes are masked or because the signal is too dim for detection. Then there are lack of available fluorescent channels in using flow cytometry. You can see the 15 colors. In confocal, you are restricted to 4 or 5, a few more now. But there is a major limitation when traditionally in the field, we've used dozens of markers to define populations of interest. This is compounded because progenitors are very rare, approximately 1 in 10,000 cells. So most of these cells are going to be differentiated cells, and you need to find the right progenitor. And as a consequence, only 1 or 2 cell types have been interrogated at a time. So, for example, the field has focused in identifying stem cells and interactions with stromal cells like vessels or the perivascular stromal cells that I was talking earlier. What has the people tried to do to solve the very soft tissue enclosed by hard tissue problem? First one is using classical cryosections. The problem of this approach was that it required either extensive decalcification up to a month in EDTA just to chelate the calcium from the bone and allowing section. And this essentially destroyed most of the epitopes, of very specialized equipment like specific tungsten blades to section the bone, coupled with a tape that you put on top of the tissue that then you peel. So these were very challenging, technically very expensive, and difficult to accomplish, compounded by loss of epitopes. Other things that people have tried to do is essentially freeze your bone and then save the surface very carefully to expose the marrow. But this also has problems of fixation. Another approaches that people have tried to do has been harvesting the bone, bisecting the bone in two, and then performing imaging, or flashing the marrow with a needle to get what we call a marrow plug, and try to keep this more or less in place to allow imaging. The problem with these approaches, and as you see from this review, is that either there is limited depth, or it requires hash clearing, loss of antigens, breaks very easily. And finally, people now with new clearing methods available have been able to clear the whole bones, but this requires the expression of endogenous reporters to visualize the cells of interest. As of today, antibodies have not been used in whole-mounted bones. What my lab uses is the bisection method. Okay, and this was originally developed in Neil Young's lab at NIH, and then used mainly by Paul Frenet lab to interrogate the localization of stem cells in the marrow. I trained with Paul Frenet, and I continue using this method. The reason that we like it is because when you butterfly the bone, you don't need fixation, you don't need any manipulation, so many of the epitopes are preserved. And this partially solves this problem, the one that many antibodies use to identify progenitors, but FACS don't work out for imaging because you preserve the epitopes that are available by not fixing or perfusing the bone. How do we do this? This is a normal mouse, black 6. You expose the chest and you can cut under the diaphragm. And you can see here the ribs. And this is the sternum in the center of the bone. You can dissect this out. Then you get this extraction, where you can see the marrow in red in this. This can get here, and then you have the top of the bone. The thymus will be just behind this. The bottom, the diaphragm will be in this position. If you get these two central segments, these are very flat, like rectangles. You can grab them with the scissors, with the tweezers, and butterfly them with a surgical blade to expose the bone marrow cavity. You essentially butterfly the sample. You can take this sample and process it like any flow cytometry sample that you have to interrogate the marrow. And this is going to do a better job of preserving the epitopes. And this is a major problem because, as you can see here, this is the bone marrow staying with anti-FLT3, either using a EPC or a CF594 conjugated antibody. And you can see that whereas 2% of the cells would be positive by FACS, by imaging, the specific signal is very low. So there is a need to preserve epitopes. To do that, we butterfly the bone and then we fix the bones in 4% PFA. And the way that we do this is we have a 96 well plate. Each well is going to have one of these butterflied bone samples. So first row is going to have PFA, second one is going to have the blocking, the stain, and then the 3 washes. Very simple approach, 4% PFA for 2 hours at 4 degrees, block for 2 hours in PBS and 20% got serum at 4 degrees, then stain for 3 hours to overnight in PBS and 1% got serum with the antibodies at the dilutions that you have to optimize for the antibodies that you use. And this is very important. Most of the antibodies that we use are directly conjugated antibodies that you buy from your regular providers for flow cytometry. BD, BioLegend, R&D, and Thermo Fisher are our main sources of antibodies. Then you wash 3 times in PBS just by taking the sample and dropping it in the next wash for 5 minutes, and then you mount and image this. For imaging, we are using Nikon AXR microscopes, but we've done similar approaches using Leica SP5 and Leica Stellaris microscopes. And this should be able to work in essentially every normal microscopy setup that you have. For mounting the sample in upright microscopes, what we do is take a plate. We are going to paint a band of super glue or gorilla glue in there. And then with a lot of care, we are going to place the sample with the endosteal surface glue into the plate and the bone marrow cavity on top. And then this is covered in PBS, and you want to do this as fast as you can, ideally less than 30 seconds. And then this is put in the microscope for imaging. For inverted microscopes, you just need to get a plate with a glass cover slip on top, integrated in the plate. You put a drop of PBS or DPBS, and then place your sample face down inside that drop. Then you take the lid of an epender, and just by capillarity, this is going to press down your sample against the glass. You can fill this reservoir with DPBS, and this is going to hold your sample in place through the whole acquisition. So how do we finish solving the other issues? The lack of available fluorescent channels are only 1 to 2 cells interrogated at that time. Well, in a paper that we published a couple of years ago, what we realized is that classically the field has used many different markers to identify the cells of interest. So for example, to identify monocyte and dendritic cell progenitors, you had to use these 2 antibodies combined with CD135 that I told you earlier doesn't really work in imaging, and then fractionate these subsets. But if you want to look at the other myeloid progenitors, in this case, granulocyte monocyte progenitors and granulocyte progenitors, you need to use 2 extra different markers. So what Jiyu, Jan, and Qingqing Gu, who led this project, did was very carefully develop new stains that allowed us to interrogate these populations of interest. And in here, we are showing you the populations that we detect using only 5 fluorescent channels. For example, you can get on CD11b negative cells and CD117 positive cells, and combine this with CD115 and Ly6C to get monocyte dendritic cell progenitors, monocyte progenitors, and granulocyte progenitors. But you then use CD115 and Ly6C again to identify monocytes and fractionate them in Ly6C high classical monocytes and Ly6C low non-classical monocytes. Then you can get the CD11b positive cells, CD115 negative cells, to identify neutrophils and neutrophil precursors, and identify pre-neutrophils, immature neutrophils, or mature neutrophils. So by carefully choosing which antibodies you use and how you combine them, you can get away from the classical approaches that require many different fluorescent channels and simplify your stains into one that now is amenable to be interrogated by using flow cytometry. Sorry, using confocal microscopy. To validate these stainings, what we do is we compare the frequencies of the cells per femur using the classical approaches or the new approaches. Then we fax purify every progenitor of interest and place it in cultures where they are going to differentiate generating colonies. And we compare the cells sorted with classical approaches and their ability to generate colonies that have granulocyte and monocyte potential, or only monocyte, or only neutrophil potential. And compare that with the new stains that we just developed and I showed you earlier. When we do these type of approaches, we find that these cells have essentially the same frequencies, telling us that we are identifying the right types of cells. Now, this is a very important. Yes.
All right, this is Diane. Just a quick question. You did that for, do you find any issues of a particular cell type having more difficulty in the fixation process that you're using? You know, I noticed that you did the granulocytes and the monocyte lineage. Do reticulocytes, or do any of those show issues about that in your methodologies?
So there are cell surface markers that don't like fixation.
Mm-hmm.
Okay, and in that case you have 2 options. Option 1 is not fixing your sample and just moving quickly through your protocol and imaging as fast as you can. And that's very doable. Or option 2, which is the one that we use in most of our studies, just to make sure that this is going to be available to everybody in the field, is to use a different marker. Right? So what we have found is that the markers that the field has classically used to identify any population of interest were generated by historical reasons. People were identifying progenitors first at CD117 positive cells, and then they were throwing more and more markers on top of these original stains to fractionate the populations. But essentially, every population in the marrow can be defined in many different ways. And when you do that, you are always going to find 1 or 2 markers that you can use, even with fixation.
Okay, thanks very much.
No problem. So a very important control when you're doing imaging of the marrow that you have to be very, very careful is that you always need to compare the frequencies that you detect by flow cytometry with those that you detect for imaging for every population of interest. And this is absolutely critical because when you are staining samples, you don't know if, as Diane was pointing out, fixation may be downregulated to markers, or you mask some epitopes. So you may have some cells that look very bright by imaging, but they might be underrepresented. You need to make sure that you are detecting every cell present in the tissue. And the way that we do that is comparing for each one the frequencies detected by FACS and imaging. What fluorochromes do we use? As I mentioned earlier, we are using a Nikon Air or Nikon AXR microscopes. These have multiple lasers that are either violet, blue, yellow, green, red, or far red. In this setting, we are using Alexa 405, BB480, and Pacific Orange from the violet laser, Alexa 488, and from the blue laser. From yellow-green lasers, we use PE, red laser Alexa 647, and far red lasers Alexa 750. The way that here I've indicated the fluorochromes that we use and the orders that we prefer to use them. In general, fluorochrome order is determined based on brightness and photostability, and especially the frequency of directly conjugated antibodies. As everybody that does flow cytometry, you know that you're not going to have problems getting antibodies in Alexa 405, 488P, or Alexa 647. It's more difficult to get BB480 and much, much more difficult to get Pacific Orange and Alexa 750 conjugated antibodies. Pacific Orange, on top of this, is very dim signal, so it should be reserved for things that are really, really bright. So when we do our stains and we finally finish the imaging, this is what we see. This is a 50 microns optical section, which is as deep as the antibodies penetrate in an overnight stain of the marrow. And you are going to see the different stains and how they look. And what we are going to do is we are going to replace each of these cell types with a dot center in the center of the cell, so that we can translate this imaging data into data that the human mind can understand. Historically, we were doing these annotations by hand, but we are moving more and more into using segmentation on software and AI, although this is not yet fully developed in the marrow. And eventually what you're going to have is a map showing the location of every cell of interest in the marrow. So in this case, for this paper, where we map myelopoiesis, what we did was stain an image of the myelogenic cells in a full bone. And for us, this was very important to make sure that we were capturing every single cell in the bone and not regions where some cells might be over and underrepresented. And I am showing you the signal for the different antibodies, plus the bone surface that we detected using second harmonic generation. We went through the sample and identified all the myeloid cells of interest and get their coordinates, and then replace them with color-coded spheres to generate the maps of the observed distribution. Now, because we have the coordinates, we can interrogate whether some cells are closer than predicted to each other, whether cells interact, whether cells cluster together. But in order to do that, we need some type of measuring stick. And what the field came up, Paul Frenet came up, was randomly place dots representing hematopoietic cells through the bone marrow cavity. The problem of that approach was that it gave you a random distribution of cells to compare with, but you were placing cells on top of vessels that I've shown you are very abundant in the bone marrow, into space occupied by vessels, or space occupied by bone, or space occupied by other hematopoietic cells. So to avoid this problem and do this in a more rigorous way, what we did is that we stain and image all the hematopoietic cells in the marrow using classical markers like CD45 and TER119 and get the coordinates of these cells. And now, because we have the coordinates of every cell in the slices of the bone marrow, we can randomly place dots representing each type of myeloid cell that we are interested in at the same frequencies found in, and we generate a map of the random distribution. And if we do this 100 times, these random distributions became normal, and you can truly compare whether two cells that are close to each other are closer than predicted from random, or farther than predicted from random, allowing you to truly interrogate cell localization in an unbiased manner.
We have a question in the chat. Sonali, do you want to ask that question? Or I can ask it. It says, are each of these markers conjugated to a fluorochrome, or were these cells expressing fluorescence like confetti mice?
So every single of these markers was an antibody detected by antibodies.
So fluorescence conjugated antibodies.
Directly conjugated antibodies.
Yeah. Okay. And then the second question that's populated is from Carolyn Auchen. I'm sorry if I mispronounced names. Is CD45 TER119 staining superior to DAPI for segmentation?
So that's an excellent question. So I'd love to be able to use DAPI for segmentation, but usually we don't have enough available channels. And then with CD45 and TER119, you need to be very careful, because TER119 stains all hematopoietic cells, including reticulocytes and red blood cells that no longer have a nucleus and cannot be detected using DAPI. And then when you use DAPI, you are also detecting all the stromal cells in the marrow, the endosteal surfaces, the endothelial cells that form the vessels, the perivascular stromal cells that are in the marrow, adipocytes, and other stromal cell types. So if you want to map just hematopoietic cells, you are restricted to using either CD45 or TER119, or using a BAF GFP reporter, which is restricted to the hematopoietic system.
Right.
In our hands, CD45 and TER119 work very, very, very well. These are very bright markers, penetrate deeply into the tissue, work really well.
Wonderful. Thank you very much.
I see that there are more questions.
I think that's it. I think they were just thank yous.
Okay. Thank you. Sorry.
No, you're good.
So how do we measure distances? So for each cell, we are going to have the coordinates of the center of the cell, but these are infinitesimally small dots. It's just 1 point in a space. So when you measure the distance between these dots, and you can use these, you can use images or you can use MATLAB of the cell to measure these distances. You're measuring from the center of each cell to the center of each other cell. You need to account for the radius of the cell. So the formula is the distance between cells is equal to the distance between the centers minus the sum of half the radius, half the diameter of cell A and half the diameter of cell B. When you are measuring distances between cell surfaces, like endothelial cells or the endosteum, you are going to create a surface with many dots in the surface, and there are many ways of doing this. My advice would be to work with your confocal microscopic core to see which approach is better depending on the signals that you use. So in this case, you only subtract the distance between the center of the cell and the Q-cell surface minus, this should be again half the diameter of cell A. Okay. So after doing all of this, what do we see in the map? And how do we visualize this? Here you're seeing the bone surfaces of a mouse sternum, and I'm showing you the localization of these granulocyte monocyte progenitors in blue, monocyte dendritic cell progenitors in green, monocyte progenitors in orange, and granulocyte progenitors in red. And each of these dots is 3 times the actual size of the cell for easier visualization. I hope that you can see that all of these cells are found as single cells, evenly distributed through the marrow, right? Telling us that we can image any region of the marrow and we are going to find these cells at the same frequencies. And we can measure the distance between them. So for example, the distance from each GMP to the closest GMP NDP monocyte progenitor or granulocyte progenitor is in most cases no different from random, with the exception of MDP, which is maybe a little bit closer when you run these very large simulations. But these cells are still hundreds of microns away on average. In contrast with, if you look at granular poiesis, what you see is and this is terminal granulocyte production occurs by differentiation of granulocyte progenitors, also called proneus into preneutrophils, immature neutrophils, and mature neutrophils that are color-coded. You can see that these granulocyte progenitors and preneutrophil clusters together. If you look at a granulocyte progenitor, there is always going to be a cluster of preneutrophils next to it. But there are mature neutrophils everywhere in the marrow with very, very, very few exceptions. And actually, these holes correspond to large vessels in the marrow. So if you were to try to infer regulation, you will find that all mature neutrophils are in contact with a granulocyte progenitor. But when you compare with random, this is not specific. Every cell in the marrow is going to be contacting a neutrophil just because of the sheer abundance of these cells. But if you compare the clustering of granulocyte progenitors and preneutrophils, you can see that they are much closer than random cells. They are in contact, they cluster together. In contrast, these immature neutrophils no longer, which are one step down in the differentiation, no longer cluster with the granulocyte progenitors, telling us that as these cells differentiate, they are leaving this structure and moving to other regions of the marrow or going into the circulation. And this is how these clusters look in real life. This is the raw data image. Now we wanted to interrogate the clonal organization of these clusters. And to do that, we use confetti mice that were developed by Hans Kieber's lab. In this model, using a global tree and one confetti allele, giving tamoxifen for 2 weeks, followed by 2 weeks rest and another 2 weeks of treatment, you are going to induce recombination and irreversible recombination and expression of either nuclear GFP, membrane CFP, cytoplasmic YFP, or cytoplasmic RFP. And this is how the marrow looks when you do that. This recombines in approximately 7% of the bone marrow cells, 3% are RFP, about 2% are YFP and CFP, and then GFP, and this is a known problem of this model, recombines a much lower frequency. So this, in theory, allows you to interrogate clonal relationships, but, and this is a known problem of the system and similar systems, is that these fluorescent proteins sit on top of the detectors that you need to use for detecting your antibodies. So, for example, RFP is going to overlap with your P detection channel, CFP is going to overlap with your 405, Alexa 405 channel, and GFP is going to overlap with your Alexa 488 channel. So how can we get away with using confetti mice? So what Ji-Yon Chin realized is that if you image at sufficient resolution, you can distinguish the fluorescent signals from cells stained with the Alexa 488 antibody for GFP or P for the RFP. Because this is a cytoplasmic signal, you can distinguish this from membrane signals. Same for nuclear versus membrane signals. So this means that if we see a cell that is GFP positive, because they have a nuclear signal, we cannot see if they also express the antibody. But if we see a cell that only has a membrane stain, we know for sure that it's not GFP positive. So this allows us to use either this channel or these channels as dumps, where we are going to put markers that we don't need to interrogate the cells that we care about. So, for example, in this setting, we are using CD115 that is not expressed in granulocyte progenitors or any neutrophil lineage cell. So when we use this trick, we can visualize now a cluster of these cells where you are going to have granulocyte progenitors that are CFP positive, that are presumably giving rise to neutrophils that are also CFP positive. Another one that is unlabeled, that is clustering with unlabeled neutrophils, and also mature neutrophils without a corresponding granulocyte progenitor. The important takeout of this is that it's possible to interrogate clonal relationships at a smaller scales. That these clusters that we see are certainly oligoclonal, and because of the organization of these structures, we think that this is a stable structure where a progenitor comes in, differentiates, produces a few cells, and then another progenitor is going to come in and continue differentiating. So to try to summarize what we found, we know that these cells are evenly distributed through the marrow, forming clusters of granulocyte progenitors and preneutrophils. When the progenitor gets exhausted, another one comes in and continues differentiating. Now, if we look at production of monocytes and dendritic cells, what you are going to see is this differentiation pathway. And again, this is color-coded in the same way. I know that this is complex, but just by looking at the mature cells, you can see that the yellow cells and the dendritic cells form unique clusters. They are not evenly distributed through the marrow. And more specifically, what we found is that these monocyte dendritic cell progenitors are going to cluster with near a cluster of non-classical monocytes on one side of the cell of the progenitor and a loose cluster of dendritic cells on the other side of the progenitor. And by comparing the distances between the MDP and classical and non-classical monocytes. The non-classical monocytes preferentially localized near this MDP. Same with the dendritic cells. So this is telling us that on top of these clusters, we have clusters of monocytes and dendritic cells around monocyte dendritic cell progenitors. And if we do confetti analysis, what we see is that there is no clonal relationship between the progenitor and the surrounding cells, telling us that either cells are recruited from elsewhere, or much more likely that the output of these cells is much lower than that of neutrophils, and that's well established in the literature. And each of these progenitors is going to generate one mature dendritic cell. We can also modify our stains and drop some channels and stain for vessels. And here you can see the maps in the marrow and where they are. And all these monocyte dendritic cell progenitors and neutrophil progenitors is specifically localized to sinusoids. You can see that even though every random cell is going to be within 5 microns of a sinusoid, these progenitors just are much closer than predicted from random, and they are actually attaching to the surface of these vessels. In contrast, they are much farther than predicted from random from the arterioles, telling us that they don't like arterioles for differentiation. And in general, they are a little farther away than predicted from the endosteum, or they don't care about the endosteum, telling us again that this is not a site for myeloid cell production. We also were able to modify our stains to visualize stem cells and progenitors and found that they are not together. So differentiation of monocytes, dendritic cells, and neutrophils is happening away from the stem cells, implying some type of migration in the marrow. And this is our summary how these cells organize. So then we ask, why is there this specific location? And looking at reanalysis of published single-cell RNA-6 from this paper from David Scadensel, what we found is that CSF1, also called as macrophage colony stimulating factor, or MCSF, which is necessary to produce monocytes and dendritic cells, was expressed in the perivascular stromal cells and in subsets of endothelial cells. And when we deleted these in the perivascular cells, we didn't find a phenotype. But when we delete CSF1 in endothelium, we found loss of monocyte dendritic cell progenitors. They were less able to generate colonies in vitro. And everything that was downstream, common dendritic cell progenitors, pre-disease, and 2 flavors of dendritic cells, were reducing this mess. And Ji Yang was able to optimize antibody staining of CSF1, which is shown here in green. And he was able to find that only some vessels express CSF1, and that the dendritic cells were preferentially localizing close to these CSF1 vessels. Some of them were on top, but some of them were also enriched in the regions around them, suggesting that these vessels were a specific niche that recruited progenitors to these structures. When we delete...
I'm sorry to interrupt, only because we're moving away from it a little bit. There was a question in the chat about how you were labeling your sinusoids.
You mean this CSF1 positive sinusoids?
I think it was a slide or two back.
How do we label this?
There.
These sinusoids. Yeah. We regularly use anti-CD31 and anti-CD144 intravascular injection.
Great. Thank you very much.
So when you deleted CSF1 in the vasculature, in endothelial cells, you see that these clusters disaggregate. The monocyte dendritic cell progenitors no longer attached to the sinusoids. The non-classical monocytes separate from each other, the dendritic cells separate from each other. So we think that the CSF1 positive vessels are a niche that recruits the progenitors and regulates their differentiation, and especially organize the production of monocytes and dendritic cells. We think that there are going to be similar niches, at least for granulocyte progenitors and probably other structures that we see in the bone marrow. And this, I hope, showcases the power of imaging to formulate the hypothesis that then you can interrogate using mouse genetics. And sorry, I made a mistake. Yes, my acknowledgements aside, I want to thank especially Yiyu Yan and Qinqing Wu, who developed the stains and the protocols to truly examine hematopoiesis. Many collaborators for this project especially highlight Cincinnati Wei, Matthew Coffron, Nathan Salomonis, Surya Prasad, Kisuke Ito, and Charles Lin, who have been indispensable for getting this to work. Our funding sources, and thank you. I'm happy to answer any other questions.
Okay, Yaping Lee, do you want to ask your question?
Hi, Daniel.
Hello.
This is Yaping, and just as I checked in the, I'm wondering what kind of microscope and analysis software did you use? The images are so amazing.
So for imaging, we use Nikon Air or Nikon AXR microscopes. This is doable in a Leica SP5 and or SP8 microscopes.
Yes.
Only that is much slower. So for software, we usually use Imaris for annotating the cells and placing the dots, and then those coordinates get exported to MATLAB. You can do this in ImageJ if you need to, right? Any regular software, image analysis software will work. And most confocal cores have this software.
Yeah. Okay, thank you so much.
Great. Other questions?
I have a hi Daniel, here's Jörg. I've fantastic talk, really impressive work. I really liked it and very educational. I learned a lot. One question, looking a little bit ahead or so. So you showed very impressively how you overcame like the multicolor imaging challenges here. And I think you had in the end what seven different colors, 7 stains, well plus then the confetti mouse stuff. Is there any need for more color ever, or is this essentially are you content with that? And if you need more colors, have you considered using these codecs or these kind of methods for multicolor imaging, or what?
Okay, so is there a need for more colors? Always. more colors allow you to ask more questions. Have we considered using the codec system or similar? We've tried very, very hard to get that to work. Okay, and I think that the limitation that there are two major limitations to the system. One is that the multiple cycles of cleaning, the bone marrow, because the cells are so loose in the marrow, they detach after you do 3-4 cycles. Then the system usually requires fixation and very harsh fixation, which means that many of the epitopes no longer work, or you only get the cells that express the most abundant epitopes. And then the other problem that you're going to have, or that we had when we started doing that, is that for good segmentation, you need 10 micron sections of the tissue, and generating 10 micron sections of the marrow is an immense technological challenge, and you is compounded by the fact that you need to decalcify, which kills even more epitopes. So this is a solvable problem, but it's going to require a lot of optimization to be able to get more colors. I think this is going to eventually work, for sure, not as well as in other tissues because of the inherent limitations of the marrow. But that if you really need to, you are going to be able to optimize some additional channels.
So we developed a new method, which we are about to like put on bioRxiv in a 2 weeks, 2-3 weeks, which should enable like this exchanging the colors in a codex manner much more easily without any harsh washes. So this might be very
yes yes
so
we'll love
and then we are currently so we are establishing this for super resolution microscopy for subcellular structures, but I think the bigger impact in the end would be for tissue level imaging the way you do it. So we are currently kind of trying to identify possible applications where we could test this. So maybe we can, I'll stay on the call here, and if they, at the end,
okay, yeah, that sounds good.
Take over this discussion here, but maybe we can chat later.
Sounds great. Other questions?
Diane, can I ask questions?
You bet. Go for it.
So, so you guys talked about the Akoya system. So the latest PhenoCycler and the PhenoImagers, you cannot do the 3D imaging that you did by confocal, correct? I think you can only do the 2-dimensional image because it's only the 10.
So for segmentation, you can only do so if you want to use their machine, you just need to use you need to generate a section.
Right.
Okay? But you can buy the antibodies from them and their conjugates and do the same type of washes that we did with their system and cleaning. So it should be feasible to do 3D. The problem is that the washes and extensive washes and fixation that the system needs is not compatible with the loose structure of the bone marrow.
Okay.
I think this is a solvable problem, and somebody that knows microfluidics and optics better than I do might be able to solve it. But at the same time, and this was very important for us, we wanted to develop tools that anyone can use in a regular confocal microscope. Right?
Yeah,
if you need to spend $100,000 up front to do an experiment, very few people are going to be able to do.
Yeah. Well, PhenoCycler doesn't have the 3D. I think the I mean, they don't have the D. Like they can.
No, it does not. But you can make one yourself. Right?
Yeah, I guess.
using low technology approaches.
Yeah. Have you done a intranuclear staining? I'm looking for a GATA2, that sort of staining with this. Is it possible to do this?
So it's certainly possible. It depends on which type of cells. So to do a nuclear stain, you're going to have to use a HARCEP fixation and permeabilization. So you need to make sure that the markers that you use to identify your cells are going to survive that treatment. That's optimization.
Right. Have you tried anything like this? I mean, in addition to, so you're just staining for the a cell surface marker.
So we've mostly focused on cell surface markers because our interest is where the cells are and how they are interacting with each other. We've imaged reporters for transcription factors, and those work very well.
Oh, okay.
In this setting, so that's very feasible.
Okay. Have you looked at the? Have you done this with the tibia or fibia or other bone marrow tissues? I just saw that this is from the sternum, correct? The section of the sternum.
This is from the sternum.
Yeah,
so you can do this in any bone. It's a lot more challenging but it's doable.
Okay, so this is not this is just the bone. Okay, so you get the section of the bone. Yeah, I've never seen so many hematopoietic stem cells in that small section. I mean, I looked at the tibia, and then we don't normally see the stem cells at all.
Well, but stem cells in the sternum, there are very few. There are between 5 and 10 per section, which is what you expect, because we are getting 50,000 cells in one of these thick optical sections. You see a lot of progenitors because we are imaging essentially every progenitor in the marrow.
Okay.
So they are more abundant, and then there are many more types.